And I guess really quickly, just since I'm recording. Attendance again, this is who is not here. Blount, Bonner, Clayton, Dawson, Ellis, Linda Q, and that's it. Everyone else is here. Also, we're a little bit behind, or I'm a little bit behind, being a little bit slow, so I'm gonna try to speed things up. Not go fast, but faster than I have been. We should have been done with this chapter already. Luckily, even though it's on here, as you guys hopefully know by now, because yesterday was the due date, none of these questions were on the exam, right? So, and they won't be obviously on the next one either. So let's move forward. Let's talk about passive transport. This was really good because this is part of what you're going to be doing in lab today, or this week, is learning about the different types of transport. Excuse me. And the first word for attendance then will be transport. So transport is the first word for attendance. If you're online, you need to send those in to be counted as present. For the rest of you, strictly extra credit. Anyway, here we go. Membranes are selectively permeable. That's an important concept, which you probably already know. But what that means is it doesn't just let anything in and out, right? It controls what comes in and what comes out, which makes sense because if it's going to maintain homeostasis, which is what we talked about way back in chapter 1, and said that was one of the properties of life, you have to be able to maintain homeostasis. It needs to control what comes in and out. If you download the PowerPoint, you could watch this little animation. It shows a little things coming in and out, and how certain things can come in and certain things cannot. Same here. It's the same kind of animation. It just looks more realistic. So you can watch that if you want. So there we go. Here's some things you already know. If you've done the pre-lab, you already know this. But here we go anyway. Molecules vibrate and wander randomly. We talked about that way back when we talked about temperature. The first time we talked about temperature, and I explained to you what it actually means when someone says, you know, the temperature in here is increasing. What that really means at the molecular level is the air molecules are moving around faster, right? As temperature increases, things move around faster. As temperature decreases, things move around less. So yeah, things are vibrating. They're always moving. And that's where we get diffusion from. First of all, you need to know what diffusion is, and that is the movement of molecules from an area of high concentration to an area of low concentration until equilibrium is reached. So basically, again, it goes from high concentration to low concentration until everything's equal. So there is no more high or low. Right? It does not require an input of energy. So some examples for that would be, you know, if I were to light a scented candle right here, first, Harrison would smell it, right? And then longer and longer it would waft out to the rest of the room, and then everybody would smell it. And at first, it would smell very strong up front and very weak in the back, but eventually equilibrium would be reached, and it would just smell equally no matter where you are around. So anyway, imagine you had a membrane separating pure water from a mixture of dyes dissolved in the water. I'll do the next slide. Actually, before I do that, this picture here, you don't need to worry about that. That's just a good demonstration of how water molecules vibrate and wander randomly. So there's a water molecule, we're all familiar with that. And you can just picture all the different ways that it can move, right? So if you can see these arrows, imagine that moving that way, kind of stretching and contracting. And then this one doing the same thing, but in alternating times. And this one kind of flapping like wings, right? You can see all the different ways, even just a water molecule moves around. Anyway, here we go. Let's talk about, I'll give you some examples of this diffusion. So imagine you put some dye in on this side, there's no dye on this side. What's happening, what we cannot see at the molecular level, is that these atoms, if you will, these molecules are bouncing around just like pool balls would, right? And eventually they're going to bounce around so much that they're going to come out of those holes. And eventually there's going to be so many on both sides. They are kind of going back and forth, but eventually you will reach an equilibrium, right? So there's about the same amount on both sides eventually. And notice, even when you hit equilibrium, there are molecules still going back and forth, right? They still move, but there's no more net movement, meaning even though there are individuals moving, generally speaking, the concentration is going to be the same on both sides. Any questions about that? All right.
Is this how our skin absorbs lotions and stuff?
Some of it, yeah. Yeah, could look at it that way. And I'm not very familiar with how skin works or lotion, but I would imagine it does get absorbed at this, at the cellular level that way. Sure, probably some of it can make it through the membrane, and some of it cannot. I would assume. But again, I really know nothing, next to nothing about the human skin system. It's a good independent question. So this next slide I'm going to show you is the same thing, but now we're just adding another color dye to it. So now you've got red dye and blue dye. And once again, you get movement until finally equilibrium is reached, at which point you still have individuals moving, individual molecules, but there is no more net movement, right? We've reached an equilibrium. As you already know from the pre-lab, the factors that influence the rate of diffusion are the concentration gradient, right? So when we say it goes from an area of high concentration to an area of low concentration, if it's, barely any difference in those concentration gradients, then it's going to be a slow diffusion. If there's one area that's really, really highly concentrated, and then the other maybe has zero concentration, that would be a high concentration gradient, right? So diffusion will happen quicker. Mass. So the bigger something is, the slower it is, generally speaking, right? So generally, if you have smaller molecules, they move faster, which makes sense because they're smaller, so they can get around things without running into other things. Temperature is another big one. Again, as you increase temperature, you increase molecular movement. So obviously, the higher the temperature, generally speaking, the faster diffusion is going to happen. And then the solvent density. So as you might imagine, like if you put something in water, it's going to diffuse a lot quicker than if you put it in, Jello, like really thick Jello. As an example. Anyway, any questions about that? All right, and once again, if you download the PowerPoint, you can watch a little animation that shows some diffusion in action. Substances that don't cross spontaneously, or really, really slowly, if not spontaneously, are transported via proteins. So again, we're still talking about diffusion, but generally speaking, a lot of things cannot just come in and out of the membrane, right? So if there was something that comes through slowly, like too slow or not at all, that's why we have these proteins. And when they use these proteins, this is something called facilitated diffusion, and you do need to know that. Facilitated diffusion. That is a type of passive transport, so it still does not require an input of energy, but it does require a protein. So say, for example, if we had, 200 people in this room, and nobody in the hallways, right? We would naturally, maybe I crank the heat up, right? And so it's just hot and miserable. Naturally, the humans in this room would want to diffuse out to the hallway, right? Naturally, we wouldn't need to put any energy. We wouldn't need to push them out. They'd be happy to get out. But the doors are shut, so they can't make it, right? Now, don't get me wrong. There's air molecules coming in and out of this room. But humans cannot get it through without opening the door, right? So you can consider the door one of those proteins. It's not that we don't need this input of energy to get us out of the room. We just need a hole big enough to get through. And that's exactly what facilitated diffusion is. Of course, sometimes it's not just the size. In that analogy, I'm talking about the size because the human is too small to fit through the crack of that door at the bottom. But sometimes it's other stuff too. There's other chemistry at play. But as far as we're concerned, there's something about these things that cannot come, or something about them that makes them where they cannot just come in and out of the membrane. Therefore, they need a passageway, and those are proteins. And that process is called facilitated diffusion. Any questions about facilitated diffusion? All right, I guess the next word for attendance will be what I was just talking about, which is door. Next word for attendance. All right, let's talk about osmosis and water balance. And once again, since you've all presumably done the pre-lab, this should be a review for you. Osmosis, the definition, at least, it's different depending on your source, slightly different. It's always saying the same thing in a different way, but your book's definition is osmosis is the diffusion of water across a selectively permeable membrane. So it's just water. It's the diffusion still, except now specifically we're talking about the diffusion of water, and it's across a selectively permeable membrane, like your cell membrane, right? If it was that easy, that would be great, but it does get a little bit more complicated as we're about to find out. Here's 3 words I've introduced to you before, and I told you would need to know them later. Here they are again. Solute, solvent, and solution, right? So when you dissolve a solute in a solvent, you end up with a solution. So when you dissolve sugar in water, the sugar is the solute, the water is the solvent, and the simple syrup is, that's what it's called, would be the solution. When you dissolve, I don't know, chocolate milk, they still make that powder for chocolate milk. Look at they do, yeah. If you dissolve that in milk, then the chocolate powder would be the solute, the milk would be the solvent, and the chocolate milk would be the solution. So on and so forth. But that is an important concept. Make sure you understand how those words are related. So any questions so far? I'm going to talk more about osmosis. Are there any questions so far? Oh, and I included this. Again, I don't make these names. I just find them out in the wild and I capture them and share them with you. But this, I've seen different versions of this, and every time I see it, I always think, oh, that's so stupid, because that's diffusion. If you're talking about knowledge going from an area of high concentration, which is the textbooks, to the area of low concentration, which is that person's brain, that would be diffusion, right? Because that's not water going from an area of high concentration to low concentration. It's something else. So anyway, yeah, when you see that meme, just know that it's incorrect. Anyway, if you download the PowerPoint, once again, you can watch a little animation. It shows all these water molecules bouncing around, and that's selectively permeable membrane. You can see osmosis in action. But here we go. Let's look at this picture. This is a classic. Nearly every textbook has some version of this when they talk about osmosis and diffusion. All right. So let's forget for a second that we're talking about osmosis. For now, let's concentrate on diffusion and also introduce you to some terms or reintroduce you. So which side here has a higher concentration of green balls? The left side or the right side?
The right side,
The which side?
right side.
Yeah, the right side has a higher concentration, right? Now, I'm going to introduce you to some terms. When we describe that in this class, when we say it has a higher concentration, we call it hypertonic. And of course, the side with the lower concentration is called hypotonic. Like, how many of you have heard, like, when someone says a kid is hyper, what do they mean by that usually? Like, oh man, that kid's hyper. What does that mean? Full of energy, right? So the kid's acting with extra energy, maybe you might say, right? So extra. So when you think of hyper, you should think of extra. Does anybody know what hypertension means?
High blood pressure.
Yeah, high blood pressure, right? So too much, right? That's hyper. Yeah. Anyway, or how many of you heard the term hypo, HYPO, allergenic? Like when someone's selling something, oh, it's a hypoallergenic dog or whatever. What does that mean when something's hypoallergenic?
It's a low chance of causing allergies.
Yeah, it's a low chance, right? So again, low you should think of as, well, hypo you should think of as low, hyper you should think of as high, and if you can remember those things, then you can understand these concepts of hypertonic versus hypotonic. All right, now again, let's pretend like we're not talking about osmosis yet. Let's pretend like we're just talking about diffusion, and let's pretend that these green balls can go back and forth across this membrane without any problem. Which way should those green balls go? What is nature going to want those green balls? Wait, do you think they're going to want to? Let's see. Choice A: are the green balls going to go this way, or choice B: the green balls going to go that way?
Choice A.
Choice A, right? Exactly. Thank you. That would be diffusion, right? Because it goes from an area of high concentration to an area of low concentration. And that would happen until we had an equal concentration, until, I don't know, I'm just making these numbers up. Let's say right now it's at, I don't know, 70%, 30%, right? And it would go until maybe you got to, that would get up to 40% as the balls started moving from left to right. And as the balls started moving from left to right, then this would decrease maybe down to 40%, or it wouldn't work. Anyway, that wouldn't work. All right. The point being, it would go until it has an equal number of whatever that equal number may be. But that's diffusion. Now, let's talk about the real situation again. Here we have a selectively permeable membrane, but the balls actually cannot go from left to right. The balls are stuck. The water can move, but the balls can't. So which way is the water going to move? A or B? Is the water going to move from left to right, or from right to left?
Water's going to move B.
Good. It's going to, yes, exactly. Water. B is the correct answer. Water's going to go that way until equilibrium is reached. So since the balls can't go back and forth to make things equal, the water will. And that's what this picture shows right here. So the water will move from left to right until equilibrium is reached. So notice the number of balls on the left side versus the right side has not changed. But what's happened is, because of the water movement, now we have less water on the left side, more water on the right side. So on the left side, it became more concentrated as the water left, or the solute didn't. And on the right side, it became less concentrated as the water came in and the solute didn't go anywhere. Make sense?
No.
No? All right, we'll do it a different way. Imagine, for some reason, this is a shot glass or some kind of drink that somebody's contracted at a bar where two people can take a shot at the same time or whatever. This obviously, let's say those green balls represent alcohol, maybe. So which side maybe would be too strong to drink? The left side or the right side? If the green balls represent alcohol. Yeah, it's too much, right? So now again, let's say you're the bartender and you can't necessarily move any alcohol around, but you do have more mixer. Which side are you gonna put the mixer on? To make them equal. You put it on the right side, right? You want to dilute it, so you put more mixer in on the right side.
Yeah.
Right? Now, if that's what you would do, then nature would do the same thing, but instead of pouring in the mixer, it would just go through that semi-permeable membrane. It's just trying to make things equal, that's all. Any other questions? All right, so everything I just said, now let's put it in writing on the screen. Everything I was just describing is something called tonicity. I'm not going to use that word. I'm not going to test you on that word, but your book uses it, so there it is. Tonicity is all the stuff that I was just explaining. Hypertonic versus hypotonic versus one that I haven't told you yet, which is isotonic. That's when everything's all the things are equal. Makes sense, right? Now, remember these are all relative terms. So if you say something is hypertonic, you have to be comparing it to something else. You don't say, oh, that's a hypertonic solution. Well, compared to what? Just like a human, right? You could say, some people might say, he's tall. But compared to what? I guess that just means taller than the average person. But then if my brother were to walk in, he'd look at me and say, he's short, because my brother's taller, right? Everything's relative. So these are relative terms. Nothing, not one thing by itself is hypertonic or hypotonic, because it has to be compared to something else. If there was one alien that showed up on Earth, and we've never seen any of the other species, we can't say that thing's tall or short, for, again, using the same example, because we don't know what the other ones look like. Of course, I guess we could say it in comparison to us, but anyway, does anybody have any questions about these terms? Again, hypertonic, there's more, hypotonic, there's less, iso, it's equal. All right, this brings us to why this matters in biology, which is something called osmoregulation, right? So this isn't just some kind of thing that you have to do in lab. Cells have to go through this, right? Cells have to deal with osmoregulation. What is osmoregulation? It is the control of water balance. Obviously, for example, you would die without water, right? We need water. Our cells need water. But as I said before with other things, there is too much of a good thing, right? So imagine if you just had water constantly coming into your cells, eventually your cells would explode, right? So for that reason, if we're talking about our cells, human cells, animal cells, generally speaking, prefer isotonic. They don't want water in there, but they don't want so much water that it's going to explode. They don't want so little water that it's going to shrivel up. They prefer an isotonic environment. So animal cells, generally speaking, prefer an isotonic environment. That is something we should know from the exam. Because, and I'm going to show you pictures of this, so it'll make a lot more sense when you get to see it. In an animal, if an animal cell is in a hypertonic solution, if the solution that it's in is in hypertonic, then the water is going to want to leave the cell and go into that solution to equal things out. And that will cause the cell to dehydrate. The opposite is true of hypotonic solutions, right? So if you have an animal cell in a solution that's hypotonic, then all the water is going to want to come in to the cell to even things out, but eventually it would lyse, which is our way of saying the cell would explode. So again, before I take questions, I have to show you a picture of what I'm talking about. So again, this is a plant cell. Either way, hypertonic, right? If it's in a hypertonic solution, the water is going to leave the cell because it wants to even things out. and the solution it's in has a lot of solute. ISO, nice and easy, everything's coming in and out at the same rate. If it's in a hypotonic solution, again, water's going to come in. Now, for plant cells, which is what we're looking at here, they have rigid cell walls. So for plant cells, hypotonic is the preferred environment. So if you remember, for animal cells, we prefer isotonic, generally speaking, and for plants, it's hypotonic. And for independent work, you could look up some examples of that, maybe some exceptions to the rule. And I don't know this, but I'm just off the top of my head, I'm thinking about what about plants that live in salt water, like mangroves, for example? What's their situation? And I don't know. If I had time, I would look it up myself. So yeah, you can look that up for independent work if you want. Any questions so far? We're still going to talk a little bit more about this. All right, as plant cells lose water, well, and as any cells lose water, they start to shrivel. But in the particular case of plants, as that happens, the plasma membrane pulls away from the cell wall, and that usually kills the plants. Not always, but usually. So here you can see the same plant. The one on the left has not been watered, but then when you water it, assuming you get to it in time before it dies, then it'll turn into what it looks like on the right. And I used to have a video where my plant looked like that. Somebody in my house forgot to water it for a full weekend. So anyway, then I watered it, took one of those stop motion videos, and you can see it go from there to there in like 30 seconds. I can't find the video though. Anyway, I don't want to take questions yet, because again, I have one more visual to show you. After everything I just said, this is what you should know, this is what you should study for the exam. Just this one slide will suffice. You need to know that animal cells prefer an isotonic environment, right? You just got water coming in and out at an equal rate. That's what animal cells prefer, because if they're in a hypotonic environment, they'll explode. If they're in a hypertonic environment, they'll shrivel up. Plant cells, on the other hand, again, they prefer the hypertonic solution. Oh, over there it is. Excuse me, hypotonic solution. So the water's coming in, kind of pushing out on the cell walls. Because again, isotonic's not very bad, but it would wilt. And of course, if it's hypertonic, it would shrivel just like animals. And again, the big ones here are knowing that the animal cells prefer isotonic, plant cells prefer hypotonic. Any questions about that? We might be able to finish this chapter today, which would be perfect. So we're done talking about passive transport. What do you think we might talk about next? See how far some of you have read it. Yes, sir. Yes, active transport. Very good. The learning objectives are this section are, by the end, you should be able to understand how electrochemical gradients affect the ions. This is according to the textbook, so we're going to talk about it, but as far as I'm concerned, we don't talk too much about the electrochemical gradient in this course. The next learning objective is you should be able to describe endocytosis, including phagocytosis, genocytosis, and receptor-mediated endocytosis. That's a lot of big words, but it's actually really simple concepts, so don't let that scare you. And then finally, you should be able to understand the process of exocytosis. And to be honest with you, as far as we're concerned in this course, the last two aren't even that important, right? But this one, even though I'm probably not going to test you on it, you need to understand what I'm about to teach you in this portion to understand how photosynthesis works, how respiration works, and that's coming later, right? So specifically, the reason for that is active transport requires energy. It's usually ATP. As far as we're concerned, it's always ATP in this class. But if you want to look it up for independent work, what other forms of energy do cells use? Usually ATP, but not always. Active transport also requires transport proteins. So just like the one we were talking about earlier, facilitated diffusion requires proteins, so does active transport. But again, active transport requires energy, unlike the one we talked about earlier. Facilitated diffusion does not require energy. Why does it require energy? Perfect. Why does it require energy? It's because we're moving solute against the concentration gradient, right? So it'd be like trying to, if you have a dam, you can just open the dam and water's naturally going to flow down because of gravity, right? But if you wanted to pump water from the bottom of the dam up to the lake, that's gonna require energy, right? So when you go against the concentration gradient, it requires energy. And why do cells do this? That's this bullet point right here. It maintains concentrations of small solutes that differ from the environmental concentrations. So for example, as you're gonna learn later, your cells need glucose, and we'll talk about that why later, but they need glucose, right? So even though they have some glucose in them, whenever they encounter glucose in their environment, they still need more of that glucose to come in, right? So they can't just depend on diffusion, because it won't diffuse, because we have to go against the concentration gradient. So any questions so far? Still have a lot more to talk about. All right, let's talk about this electrochemical gradient. Basically, and I'm not going to ask you this, but the interior of the cell is negative relative to the exterior. So your cells actually have a charge. Generally speaking, the inside is negative relative to the exterior. And again, I'm not going to ask you that, but now you know. So long story short, there are reasons other than the concentration gradient that necessitate active transport. It's the only reason I even included this, well, that because it's in the textbook. So I'm not going to ask you about the electrochemical gradient. I just want you to know it exists. And even though we will be focusing on the concentration gradient, I just wanted to throw it out there and make sure we recognize what the textbook says. And yes, there are other things at play, but for the most part, they won't matter for us. Like even later, we're going to learn about, there's going to be a very important process that involves hydrogen ions going against the concentration gradient. And hydrogen ions are charged. So that does play into it. But even then, we're not even going to focus on the electrochemical gradient of that process. We're just going to focus on the easy part, which is we've got hydrogen ions, a lot of them here, not a lot of them there, and we're going to pump them against the concentration gradient. You'll see what I mean when we get there. Anyway, any questions about the electrochemical gradient?
Is that why we can touch our cell phone with our fingers? Because we're negative charge?
I don't know. And I don't think we are necessarily negatively charged. It's just saying the inside of the cell relative to the outside. So it could be that we balance out, you know. If you look at the inside of the cell, you're like, oh, that's relatively negative. Then look at the outside of the cell, it's like, oh, that's relatively positive. And what happens when you have negative and positive together? They generally balance out.
Cells balance out.
But that's another great question too, and I have no idea how iPhones work or touch screen. I'd be interested. Like, is it pressure? Is it temperature? I don't know. Another good independent we're talking. And that's barely, right? That's normally, since we've said it, we can talk about it, but because that's almost technology and not biology. But we are talking about figures, so how does that work? You can look it up if you want. Anyway, download the PowerPoint, you can see this animation that again shows active transport in action. And in this case, they even talk about a very famous one, and your book does too, but I'm not going to ask you about it, the sodium potassium pump. You see sodiums have a plus charge, the potassium have a plus charge, and that all falls into play with not just the concentration gradient, but the electrochemical gradient and a bunch of stuff you don't necessarily need to worry about. The point here is we're moving things against the concentration gradient, and it's going to require proteins like that one and energy, because this is active transport. So again, a picture of it in action. Whatever this solute is, you can see there's 4 of them on this side of the cell. There's only 1 on this side of the cell. So diffusion would actually make it want to go that way, but we don't want that. Whatever this solute is, the cell wants more of it. So it has to go against the concentration gradient. So it comes in this protein, and we have some energy, and then it lets it in, right? That is active transport. As far as we're concerned, it's pretty simple. Now, if you were to really get into it, it's actually pretty complicated. The only thing you need to know is sometimes we need things to go against the concentration gradient. Do this with proteins and energy, and the whole process is called active transport. So any questions about that? Also, these pictures are from the old textbook, not your current textbook. I thought the old textbook actually had better pictures, so I kept them in here. But you have access to both, right? You can look at whichever pretty pictures you want. So a new thing that we're talking about, a different thing we're talking about, is endocytosis. So again, this is also a type of active transport. But usually when any 100 level textbook talks about this, even though they do say it's an active transport, they don't really focus on the fact that it needs energy. So I guess just keep it in the back of your mind, this does require energy, but for some reason we don't focus on how. But anyway, endocytosis is a type of active transport. What is it? It's when cells take in material. That's basically what you need to know. Yes, it buds, when a vesicle buds inward. I'm not gonna ask you that. If anything, you just need to know that endocytosis is when a bunch of stuff is coming into the cell. So we're not talking about like one thing at a time where you can use a protein like we've been talking about. We're talking about a bunch of stuff at once. And then you need to know this. Endocytosis is broken down into two subcategories, phagocytosis and pinocytosis. So one of them is for solid stuff coming in, and the other one is for liquid stuff coming in. How many of you know what, I don't know, Pinot Noir or Pinot Grigio is? What's that I just described? What? Sorry, I couldn't hear you.
Alcohol.
Alcohol, yeah, it's a wine, right? So when I was a student, that's how I remembered Pinot is drinking, right? So when I just remembered that a Pinot, different types of wine, you have to drink wine. And then by process of elimination, the other one must be eating. So any questions about that? We'll make a good time. So if that's endocytosis, the opposite is something called exocytosis. And it's just, instead of things coming into the cell, it's stuff moving out of the cell. And once again, it's a lot of stuff at once. This is why we can't just use proteins or simple diffusion, right? Because we're dealing with a lot of stuff. In your book, give some examples. I didn't include them because I'm not going to test you on the examples. But again, for independent work, you can look this up. Some examples of when cells use endocytosis, like why, in one situation, are they using that instead of active instead of simple diffusion or facilitated diffusion. Anyway, any questions about endocytosis or exocytosis? All right, download the PowerPoint, watch this little animation. And let me at least pull the next chapter up and we'll start the introduction. That'll be it for today. So I don't mind getting ahead, starting later, ending early today. Because I've got a lot of grading to do. Bear with me while I pull this up. Does anybody have any questions while I'm pulling this up? Everybody's good on the pre-labs? And again, if you haven't done the pre-lab, do that as soon as you're done with class, if possible. And don't forget, this one involves a video that you have to do, a video and questions again, and it also involves hypothesis questions. There we go. All right, sorry about that. In this lab, just so you guys know, this week will be one of those, another team effort one. So be ready to be working with the team. You guys will be split up. So it won't be like one huge team. It'll be group one does this, group two does this. And as one of your classmates recommended for the last lab, she thought that you guys should switch around, like you do this experiment and then go on to do the next one. And I love that. In a perfect world, is exactly what we would do. But I know for a fact from past experience that there's not enough time to do that in the lab. So while you're doing your experiments, please feel free to walk around and see what the other people are doing, because even though you're not doing those experiments, as you know, you are responsible for those questions, right? And getting that data. All right, finally it's downloaded. Once it's downloaded, we can get started again. All right, the next word for attendance is waiting, since we are waiting for it to download. There we go. As Tom Petty would tell you, the waiting is the hardest part. Actually, that's not true, is it? Not in this situation. It's easier to wait for the PowerPoint than have to digest it sometimes. All right, really quickly, this is a big one. This is an important chapter that we're about to cover. How cells obtain energy. It's going to be very important. We're going to do a lot of stuff on the exam about this. And in my opinion, probably one of the harder concepts is this next exam. The exam 2 is going to be basically all about this. It's all about energy, whether it's plant cells getting energy or animal cells getting energy. It's going to be all about energy. Historically, it is one of the hardest. So just a heads up, try to make it easy as possible. The first thing we're going to talk about, we can get into this, energy and metabolism. Like before we even talk about how cells get energy, let's first talk about what energy actually is. So the learning objectives of this section are, at the end, you should be able to explain what metabolic pathways are. You should be able to state the first and second laws of thermodynamics. You should be able to explain the difference between kinetic and potential energy, and I bet most of you already can. You should be able to describe endergonic and exergonic reactions, and I'll go ahead and tell you now, you will need to describe them, but I'm not necessarily going to be focused on the nomenclature, like endergonic and exergonic. I'm not worried about the words. I just want you to understand the concepts. And then finally, you should be able to discuss how enzymes function as molecular catalysts. And if you don't know what a catalyst is, that's something that basically helps a chemical reaction happen. All right, the first word not attendance, but the first word from your textbook in this chapter is bioenergetics. And I'm not going to ask you to define it. Let's talk about it anyway, because even though I'm not going to ask you to define it, this is basically what we're going to be talking a lot about. Bioenergetics describes the concepts of energy flow through living systems. And I made this analogy before, I don't remember when, but I did, way back in chapter 1 when we said the ability to transform energy from one form to another is one of those things that it's a property of life. And then I mentioned, like when we need energy, when humans need energy, we can't just go outside and, in the nude and absorb all the sun energy and then we're good, right? That doesn't work. We have to eat. But even when we eat, that energy did ultimately come from the sun, right? If you eat a steak, all the energy in that cow came from all the grass that it ate, or hay, or whatever plant it ate. And all that energy in the plant came from the sun, right? So that's where the sun energy ultimately comes from. Here's another term that you actually do need to know, though, this third bullet point right here. All chemical reactions that take place inside cells is known as metabolism. So all the chemical reactions happening inside of you right now, and there's a ton of them because that's what's keeping you alive, is chemical reactions. That is called metabolism. That is what metabolism is. So any questions about this? All right, there's two different types of metabolic pathways you need to know. There's anabolic and catabolic. Anabolic needs energy because you're basically building a new molecule and it takes energy to build something. Catabolic produces its energy because you're breaking something. When you break something, and I'm going to talk more about this later, this content. When you break something, whatever energy was holding it together is then released. So make sure you understand the differences between anabolic and catabolic. And again, later we'll talk about why that happens. Like why is it that it requires energy to build something? Why is it that when you break something, it releases energy? We'll talk about that. But for now, just know anabolic requires energy, you're building something, and catabolic is when you're breaking it down. Like how many of you have heard of anabolic steroids? Right? And what's that? Well, let me ask you, what's it abused for? I mean, forget the actual medical use. How do people abuse anabolic steroids? What do they use it for? Yeah, to build mass, right? They're building mass. And literally at the cellular level, they're building new molecules, and that requires energy. Anyway, again, we'll talk more about those concepts in later in this chapter. Both of them require enzymes to happen. We're going to talk a lot about enzymes in this chapter. So for now, are there any questions about this before we come back to it? Okay. Let's talk about thermodynamics. That is the study of energy and the energy transfer involving physical matter. I'm going to put a question mark next to this indicating, not sure if I'm going to ask you that on the exam or not. But if thermodynamics is the study of energy and energy transfer, then what is energy? And this is another word. I'm not going to put a question mark next to this. You do need to know this one. Energy is the ability to do work or to create some kind of change. So there's a lot of different examples. You don't need to know any of these. You can figure out and write some more if you can think of it for independent work. But electrical energy, right? Heat energy, light energy, chemical energy, sound energy, right? There's all kinds of different energy. Are there any questions so far? All right, back to thermodynamics. Here's something you need to know. The first law of thermodynamics states that energy can be transferred from one form to another, but it cannot be created or destroyed. Now, this is the, let me think. That definition verbatim is, I think, from the old textbook. So if you were to look it up in your textbook, it would be worded differently, but it's describing the same thing. Just in case you're like really a stickler on the textbook versus the PowerPoints. So as you might imagine, life, again, not imagine, I told you this is one of the properties of life. Life depends on the conversion of energy from one form to another. Right? This is what this whole exam is about. Exam 2 is all about that one property of life, transforming energy from one form to another. Which brings us to the second law of thermodynamics, which states that energy is lost as it transfers from one form to another. I have a typo there. But yes, so you need to know that. The first law of thermodynamics, energy can't be created or destroyed, right? It can only be transferred from one form to another. And hey, by the way, while we're talking about that, the second law, when that happens, you always lose some energy. Always. Anyway, any questions about that? All right. We've already talked about this. This first part should be a review for you. We've mentioned this a few times. Heat is a type of energy. You need to know that. Heat is a type of energy. Specifically, it's a type of kinetic energy, which is the energy of movement. And heat is the random motion of matter. So that's how your book is defining heat, the random motion of matter. So again, I've been using this example for a long time. When I say the air is getting hotter in here, what I mean is the molecules are moving more. Heat is also the most chaotic / disordered form of energy. As far as we're concerned, that doesn't matter. If we were doing this properly and using mathematics, that would be an important bullet point, but in this case, it's not. And this one, I don't know, I'm not going to X this one out. But all energy conversions generate some heat. So every time you go from one form to another, some heat is generated. And again, if we were doing this mathematically, which we're not, that would be important. We would actually need to mathematically account for the heat and account for the disorder. But in this case, we don't need to. So the most important thing you need to know is that heat is a type of kinetic energy, and it is the random motion of matter. So make sure you know that second bullet point. Don't worry, actually the first bullet point, don't worry about the second one and the third one. It's good to know, but it's not very important in this course. And that is the same thing I can say for all of entropy. I'm going to define it for you. Entropy is the measure of disorder or randomness in the system. And I will tell you that every time energy is converted from one form to another, entropy increases. But now that I've told you that, because it's in the textbook, I'm going to tell you go ahead and brain dump it if you want. Entropy is a very important concept when you're doing the math on this. That has to be accounted for when you're doing the math. We're not doing the math. So if anything, you just need to know that entropy is just a measure of chaos. And as you go from one form to another, you increase it. So here's a good picture. You can see all these things highly ordered, nice and neat, but then they've diffused away from each other. So there's more entropy, more chaos to the right. And again, that's a very important concept for people who are doing this math, but for us, it's not. The textbook included it, so did I. There you go. Any questions about entropy or heat? All right, moving right along. I've already brought this, used this topic, this word. I've already used it today, but let's just go ahead and define it. You probably already know it. Kinetic energy. It's the energy of motion, right? That's why when we said heat is a type of kinetic energy, because that's a type of, it's energy of motion. It's the molecules moving around. That's what kinetic energy is. And then you have potential energy, which is stored energy, right? So kinetic is the energy of motion. Potential energy is stored energy. How many of you, I'm not going to ask that, but I would assume most, probably all of you knew those two definitions, right? Kinetic energy, energy of motion, potential energy is stored energy. Here's where it gets a little bit more complicated. They don't always say this when they teach you about potential energy, but yes, these are important concepts too. That stored energy could be from location or structure. And usually, like science classes in high school, usually focus on the location part. Like they'll say, all right, which one of these has more potential energy? The red one or the black one? The red one, right? Because it's higher, right? They always teach you that. And that is true. But for biology, that's damn near useless. What's more important for us, and they don't usually teach this a lot in high school, I don't think, is the structure part. And this is what I was getting at earlier, and I'm gonna talk a lot about it later. So it takes energy to build something, right? And however much energy it takes to build something, to build that structure, that's how much energy is ready to be released when you break that structure. When you break those chemical bonds, break the structure, that's what the potential energy is. So again, as far as we're concerned, in this class, the potential energy we're worried about is the potential energy stored due to its structure, or more specifically, those chemical bonds. And again, we'll talk about that in more detail later. Here's a great picture. It's a great example, right? There's a lot of potential energy in this dam, right? There's a lot of water dammed up right there, right? That's a lot of potential energy. And here you have this waterfall, and that water is moving. So on the left is an example of potential energy, and on the right is an example of kinetic energy. And let's just look at this one picture before we're done, I think. Yeah, almost. This is, again, a picture from your old textbook, and it's a decent way of putting it, right? As these people are having their fun, this is the least amount of potential energy they will have while they're doing this, right? And they climb up, and as they get higher and higher, by the way, it's taking kinetic energy to get up there, but the higher they get, the more potential energy they get. In this particular case, since they're not flying up into the sky, this is the greatest amount of potential energy they're going to have as they're playing. Then when they dive, right, that potential energy is then converted into kinetic energy as that person moves. And then they hit the water and you can hear it, and that's actually a type of kinetic energy too, because sound energy is nothing but vibrations in the air, which again is movement. If you download the PowerPoint, you can watch this little animation that talks about what I'm going to talk about when we come back on Wednesday, which is chemical energy. So that's it for today. If you have done the pre-labs, make sure you do them, and I will be online for all those hours that you need. Have a great week.